All right, so let's start to talk about how we study cells and how we use cells. All right, so we probably know that microbiology is the study of things too small to be seen. So almost everything we do with biotechnology involves cells. Right, so the traditional things for microbiology would be like bacteria, most common, and protists, which are single-celled animals, usually living in aquatic environments. Yeast, we use yeast quite a bit for biotechnology. Algae, fungi, we use fungi quite a bit. And then, instead of protists, I'll say eukaryotic cells. Right, so just basically eukaryotic cells. And it'd be like us, we're eukaryotes. Right, so we already talked about prokaryotes, those are bacteria. And then everything with a true nucleus is going to be eukaryote. So the field of biotechnology, a lot of companies like Amgen's are big, or a big biotech company in our backyard. They're up in Thousand Oaks area. Amgen started from a bunch of guys who made beer, right? So they used yeast, and they were sciencey guys who said, hey, you know what? We could take our knowledge of how we culture yeast and use it to make cultures of bacteria, and those bacteria could produce medicines for us. So interesting how they started, but that's how they started. And now they're a multi-billion-dollar company. So microbiology is the study of things too small to be seen. Like I said, organisms would have to be viewed with a microscope. So a couple things we'll get out of the way. Manipulating a bacteria is called genetic engineering. So we'll start with the simple stuff first. Remember we said that through plasmid manipulation, right, we can transfer genes. So in plasmid manipulation, so genes, genes are inserted via restriction sites. So a restriction site is a restriction enzyme is an enzyme that cuts DNA. So it cuts DNA. We engineer these restriction sites to be complementary or to match the DNA that I want to stick in there. Now we're going to talk about that much more in the next chapter. But we can engineer these restriction sites inside plasmids. And then we can get these plasmids to upload into bacteria, and then they can produce stuff. So a couple things we've been able to do, we make what are referred to as naked E. coli. So they're engineered to be non-pathogenic, like our HB101K12, right? And they're also engineered to have multiple restriction sites. So I don't have to go find E. coli and then hopefully it does what it's supposed to do and it's not going to hurt people. We have whole strains of bacteria that are genetically engineered. Right, so adding a gene makes it a recombinant DNA. Right, so recombinant means DNA of two different organisms. All right. Having foreign DNA, that's what recombinant means. Man, that's just not good, right? Let me see if I can rewrite that. Make it pretty. Oh, yeah, having foreign DNA, that's what recombinant means. So, for instance, what we would have been doing is we would have been transforming bacteria now, making what's called pea glow, right? Or green glowing bacteria, right? So normally bacteria would not grow green, but we can insert a fluorescent protein in it, and oftentimes we'll put these fluorescent markers in our cells so that we can identify ones that have been transformed properly. Other cell types we use, so remember a biologic? That's a medicine. Right, it's a medicine made by a living cell as a biologic. So a lot of things like Tylenol or Advil, those are chemicals, right? So they're made in chemical plants by just mixing things together and combining them. But a biologic is a protein that has to be folded, right? So if it requires minimal post-translational modification, right? So remember bacteria, so if I have a bacterial cell, right, it'll have its one circular piece of DNA, right? And it'll have a bunch of ribosomes that are all around the DNA in the nuclear region. And what happens is, when the RNA starts reading the DNA, it's made right at the ribosomes. And it will fold naturally based on polarity and whatnot. But it doesn't get modified much, right? So if we think about a eukaryotic cell, right? So a eukaryotic cell, I have the cell, right? I have the nucleus, and in the nucleus I have lots of DNA, right? But outside the nucleus, I'll have the endoplasmic reticulum, right? And then I'll have the Golgi. So I have all these chants, and I have all my ribosomes on my endoplasmic reticulum. But I'll make my proteins on the ER, or the rough ER, and then I'll send it to the Golgi, and it'll be modified further. So in eukaryotic cells, I have all this chance for modification. Right. So if it requires minimal, then I'll use bacteria. Because bacteria is just going to fold based on polarity. An example of that would be insulin. Insulin is a protein produced in the pancreas. We use it to bring sugar into cells. It's great for diabetics, but it requires very little modification, so I can make it with bacteria. Right? Biologics requiring minimal modification still. I can use fungus. Right? Aspergillus is black bread mold. We use it to make a lot of detergents. Right? So the folding has to be a little better, but it doesn't have to be great. Right? I can use an armyworm. Right? And those cells you can buy in culture, and they'll make a little more modification, a little more folding. Remember, the folding of the conformation, right, gives function, right? And then I can use yeast, right? Yeast are single-celled eukaryotic cells that we use to make, beer and bread and stuff like that. But they'll do a little better modification. But the problem with this, right? This is an insect, right? And this is it's a mold, right? This is a mold, right? Or actually, yeast is a fungi. Aspergillus is a mold. But they're not human cells. All right, so if I need a lot of post-translational modification, I'll use a mammalian cell. So Chinese hamster ovaries, or CHOs, are cell lines that have been around for a long time. They've been modified. And they can do what's called extensive glycosylation. So they have the whole modification pathway, and they'll make a mammalian protein, which is really similar to what our protein would be. So we can program these to do more or less glycosylation, and then it makes the biologic much more acceptable to humans. Ultimately, we'd love to use stem cells, right? So stem cells, remember, have the ability to become other things, right? So there's three main classifications, right? So there's totipotent. Totipotent can become a whole organism. So when I have a new fertilization, right? It starts with 1, it makes 2, it makes 4, makes 8, right? Up till about 32 cells, right? All of these are totipotent. Right, so these cells can become anything. But the question is, where do you get these cells to do research, and where do you get them for large-scale production? Right? The problem with the totipotent cell is each one of these cells here, each one of these cells can become a person. Right? So there's a lot of ethical issues surrounding it. We have, if you think about in vitro fertilization, right? So they induce a woman to produce a lot of eggs, and they fertilize all these eggs, and they freeze all these, right? And they'll grow them to this point, to these 32 cell point, right? And they'll rest them, they'll freeze them. And they'll stop their development. And they'll, if they've got 8 cells, they may implant 3 or 5. But then they have 3 left over. So these cryobanks, these frozen cell banks, had all these embryos, right? And then scientists started saying, well, if you're going to throw them away, if you're going to dispose of them, just give them to us, right? Because we can do research. Well, the Bush administration got wind of this, and they put a halt to it, because the problem is each one can become a person. And based on what you believe, right? Some cultures don't believe that people are people until after a year after birth, and some people believe that it's at fertilization. So we put a stop to that. Pluripotent cells, right? It can become any one of any cell in a certain tissue type, right? So if we have connective tissue, right, a pluripotent cell or an IPS, right, can become any one of a whole bunch of different cells, right? So it can become a liver cell, a skin cell, right, a heart cell. So any one of these, it can become. So pluripotent cells are important too. So once I pass this 32 stage, once I get to 64 cells and older, so these become pluripotent cells. So those become pluripotent cells, right? So the problem is totipotent and pluripotent, and both of those require that I harvest embryos to get them. You don't have pluripotent cells, right? You have a type of pluripotent cell that in biotechnology we call multipotent, right? So you have, in bone marrow, you have a type of pluripotent cell called multipotent cell, and that can become any type of blood cell, right? So, but what we found out, right, through biotechnology is I can make a multipotent cell into a pluripotent, right? So a couple things happened. So these ones were banned. Those were banned in the 1990s. But President Obama, when he was president, said, well, we can use the existing ones we have. So they were reopened in the early 2000s, right, 2000 up to 2010, but there's a limited supply. But now I can make pluripotent cells from multipotent cells. And then we can try to establish cell lines from those. So obviously, if I could make iPSCs to make proteins, that would be better because they'd be human in nature. So once I figure out the cell I'm going to use, I create a cell line for study. That's called a cell culture, right? So we use them to develop and test biologics. So plates must be specific for the organism. So every organism has different requirements. Like fungus grows better at 30 degrees Celsius, and mammalian cells like the CHOs grow better at 37 degrees Celsius. The pH might be slightly different. The gas requirements might be different. The growth factors might be different. So each one is grown at a different one. This red color is common, and it's phenol red, and it changes to red at a certain pH. Right? So when it starts to change color, we know that our pH is off. Right? Often we'll give them an antibiotic resistant. We need to keep our cell culture sterile. Right? So if anything infects it, the whole thing is trash. We have to throw it away. Right? So we have to make sure that we use aseptic technique. We'll talk about that in a second. And then we have different ways of studying. Right? Cell fractionation is splitting cells. Right? So they're split using enzymes so we can harvest the parts. Right? They're separated with centrifugation. Right? So I'll use cell fractionation to split them. Homogenation means mixing, like with a blender, and then centrifugation will separate the parts. Right? The heaviest parts go to the bottom, and the lightest parts stay on the top. So, when centrifugation, we'll talk about it. The heaviest part will make what's called the pellet, and a lot of times what we're interested in is in the supernatant, or the goo that's on the top. Right? All right, and then we get to cell culture. Right? So, cell culture had its own kind of challenges, right? So, in small scale, so when I started cell culture, I could use a T-tube. So, in a T-tube, so in your body, cells usually will only grow when they're attached to something. That's called anchorage dependence. And they'll grow until they meet another cell and they'll stop. Right? That's called density dependence. And it's a safeguard to help prevent cancers. Right? So when we start cells, and we may start them in tea flasks, right? But there's a problem with that. I can't go large scale with that. And I'm limited to the space that cells can grow across in a tissue. Right? Once I'm able to do this well, then I can induce these cells to give up these anchorage dependence and density dependence. And I can engineer these cells. A lot of the CHO cells are engineered to give up their anchorage dependence and density dependence. And I can grow them in liquid cultures. Right? So a liquid culture, I could use a spinner flask. Right here. So this has a propeller at the bottom, right, or an impeller, right? And it will spin and it will mix it up. It will keep everything mixed up in culture, right? And then I can pump gas in here, and I can remove stuff. So I can hook different tubing systems up to maintain the internal environment. So that's a small scale. I can use roller bottles. So roller bottles, kind of like a spinner flask. But these bottles will be on heated rollers, and they'll just continue to roll and cycle around. Right? That's called a roller bottle. And then I can scale up to what's called a bioreactor. So a bioreactor, it's just like a spinner flask, but on a large scale. Right? So I'll have a reactor tank, I'll have a thermal jacket, I'll have lots of sensors, I have a way to pump gas in, remove gas out. I can remove effluent on the bottom. That's the stuff I want to remove from it. I can put new medium at the top. And then bioreactors are larger scale. So this is a bioreactor right here. Right? So we have larger ones, we have smaller ones. And they have some bioreactors that go up to 2-story buildings, right, when I'm up in production. So different ways we can do it. This is just an introduction. You should take a look at your book. It's got a lot of good information in it. And I will talk to you soon. Okay?